And with that, I would love to introduce Susan Gilliland, who will actually introduce our speaker for the night. Susan.
Okay, thanks, Ron. Well, tonight we have the great pleasure to welcome Dr. Myra Finkelstein. Myra is a native Californian and received her undergraduate degree from UC Davis. Before returning to graduate school, she volunteered with several nonprofit organizations working on conservation issues, mostly marine conservation. But she says she always felt a little bit limited because she didn't have a good understanding of effective solutions. So Myra took graduate courses at UC Santa Cruz, studying aquatic chemistry and physical oceanography. She also started volunteering in a lab that measured contaminants, such as organic chlorines and trace metals. Well, the rest, as they say, is history. Myra earned her PhD in environmental toxicology, and today she's an adjunct professor in the microbiology and environmental toxicology department at UC Santa Cruz, where she lives with her young family. Myra's research focuses on human impacts to wildlife with an emphasis on contaminant-induced effects. Dr. Finkelstein and her team at the UC Santa Cruz were the first to definitively link ammunition to the lead poisoning impeding the recovery of free-flying California Condor populations. She and her colleagues first assessed the frequency of lead poisoning as a cause of the illness and death in the birds, and then showed that lead in the ammunition had the same chemical signature as that in the bird's feathers. Myra and her team's research played a significant role in raising awareness of the lead ammunition issue, which resulted in the passage of the bill to prohibit the use of lead ammunition for hunting throughout California, the first such law in the nation. Dr. Finkelstein has also focused her research on contaminant-induced effects and toxicology in marine seabirds, such as the black-footed and the Laysan albatross, which is her personal favorite bird, where she uses biomarkers of contaminant exposure to examine the population level changes on these seabird species due to marine pollution. Dr. Finkelstein's work is part of a new hybrid of research using environmental toxicology to inform conservation biology. So please welcome Dr. Myra Finkelstein.
Thank you. I will share my screen. Okay, thank you very much, Susan, and thank you to the LA Birders. It's my pleasure to be here today. I'm going to talk to you about some of my research and specifically the work that I do, which I call conservation toxicology, which, as Susan said, it's a hybrid of environmental toxicology and conservation biology. But first, I just want to talk to you a little bit about lead, and I'm just going to leave this up and give people a chance to read this for themselves. So this quote was from Orfila, which is who's known as the father of toxicology over 200 years ago. So to me, it's amazing that for over 200 years, lead has been, basically known as such a well-known harmful toxic compound. And indeed, lead is toxic across multiple physiological systems, immune systems, reproductive systems, neurological systems, across vertebrate species, and lead is toxic to children with no safe level of exposure. So lead is very toxic, and we know it's very toxic, and we've known it's been very toxic for over 200 years, and it's still such a problem today. So I'm going to talk to you about specifically generating information to help reduce lead poisoning in avian species. And I'm going to talk to you about this framework that I use, which is to try to understand the problem, quantify the impact, and help identify solutions for the problem. And I'm going to walk you through two case studies, one with Laysan albatross and the next with California condors. But first, science is a collaborative effort. And so I'm going to talk today about this work that some of it we've generated in my lab, but that has been a collaborative effort across the board. I have multiple current collaborators from different universities and as well as agencies such as the Fish and Wildlife Service, National Park Service, and as well as nonprofit organizations. So although I'm giving the talk, it's not just me that's done this work. I think it's important to show that. So I'm going to first talk about understanding the problem, quantifying the impact, and identifying a solution for Laysan albatross. So they're the smallest North Pacific albatross with a wingspan of about 6.5 feet and weigh 6 pounds. They can live up to 60 years or longer, which you'll see in just a second. They start breeding at about 8 years of age, and they have about 1 chick per year or 2 chicks every 3 years. So what we consider low reproduction. But one of the major stars of the Laysan albatross world is Wisdom. And I know a few of you have heard of her. So she has her own Wikipedia page. She's the oldest confirmed wild bird in the world and the oldest banded bird in the world. And I just looked up, I do this every year to see how she's doing. And just this past March, she's hatched another chick and she's at least 70 years old. Amazing. And I love also, like, I'm not on Facebook, but Wisdom is. She has her own Facebook page also. So for those of you that are on Facebook, you should follow her. She's a pretty cool bird. So the work I'm going to talk to you about today took place on Midway Atoll. It's the middle of the Pacific Ocean here. And hopefully you guys can see my thing a little bit. It's currently U.S. Fish and Wildlife Refuge, and it's a former military base. And this is a picture of Sand Island, Midway Atoll. It has the world's largest breeding population of Laysan albatross, about 70% of the global population, and about 400,000 pairs. So it's a really important breeding ground for Laysan albatross. And I was fortunate enough to do my PhD work on Midway. And when I went to Midway to study Laysan and Black-footed Albatross, I saw this. So this is a Laysan albatross chick with something that we call droop wing. So basically this bird's wings are paralyzed. It's peripheral neuropathy. So this bird will never be able to fly. So it's a dead bird. So when the parent stops feeding this bird at the end of the breeding season, the chick will die because it can't fly from the island. So it's really sad to see. And I saw all these kind of Laysan albatross chicks wandering around with these droop wings when I was out there. And I immediately thought of this. So now this is a picture of a painter, and it's a painter with acute lead poisoning, and he has something called wrist drop, which is also a sign of peripheral neuropathy. And so I was worried whether or not these Laysan albatross chicks were also lead poisoned, and if lead poisoning was causing this peripheral neuropathy, similar to what you see in humans. So the first thing I did is I took blood samples of Laysan albatross chicks that nested near the buildings here, and also chicks that were far away from the buildings, because we had a feeling that the lead-based paint from these buildings might be impacting the chicks. And so on the y-axis is the blood lead, and then near buildings and away from buildings. And I found that by and large, chicks near the buildings had astronomically higher blood lead than chicks away from the buildings, with about 90% of chicks near the buildings having this blood lead that could trigger this condition we call droop wing, which was basically a sentence to death. So it was a lethal exposure level, how you will. So instead of just saying, well, we think it, well, trying to figure out, well, what could be the plausible sources of lead exposure to these chicks? So now here on the y-axis, I have blood lead, but it's in log scale, with chicks near the buildings, chicks far from the buildings, and then adults, which are really low. So what could be sources of lead to the chicks? Could it be diet? Could it be something that the parents are eating at sea and bringing back to the chicks? Well, no, that's really not likely because the adults are so low. But Midway is a former military base. And when actually I was out there doing this research, people told me, well, you know, Midway is very contaminated. You know, the Battle of Midway took place here. There's lots and lots of contamination around the island, could be all over the place. So that was something that was proposed that could be what's poisoning the chicks, except for it seemed unlikely because the chicks that were far from the buildings didn't have high lead. So it wasn't this sort of ubiquitous contamination throughout the island. It was limited to these chicks that were nesting near the buildings. And lo and behold, these buildings that the chicks were nesting near were painted with lead-based paint when the military occupied the island. So they're these old peeling, lead-based paint peeling buildings, and the chicks with the droopwing would have nests right next to these peeling buildings. So it seemed likely that lead-based paint was probably the source of poisoning the chicks. But to actually determine that definitively, we could use stable lead isotopic analysis. And you can do this because lead exists as 4 stable isotopes, and the lead signatures in the organism reflect the lead signatures in the source. So example, if I wanted to eat a whole bunch of lead-based paint, the signature of the lead-based paint would basically, that was in the paint, would match the signature in my lead poison blood. So you don't see a change. But you need highly sensitive instruments to measure the different isotopes of lead, and you need to do it in trace metal clean techniques, so you're not adding external contamination. So we work in a class 100 air clean room, and wear booties, and very, very careful about external contamination. But then I can analyze these samples. So I went around, and here's another droop-winged lead poison chick, and I pulled paint chips out of the chick's nest. And I also took a blood sample from the chick, and I measured the lead ratio in the paint and the lead ratio in the blood. On the y-axis here is just the lead ratio. And then you can just think of it sort of like a fingerprint, but not quite as unique. And I did this for multiple chicks on the island. I'm just going to show you a handful. But in every case, the lead isotopic signature in the paint from the nest matched the lead isotopic signature in the lead poisoned bird's blood. And so showing that the lead paint was the source of the poisoning to the chicks. And you can see here, like from chick 1 and chick 2, that the paint is very different isotopically. These buildings have been painted over decades with different types of paint. So every paint on every building didn't match, but all of the chips from the chick's nest matched that chick's blood. And it was amazing to be working on Midway, and a bit, what's the word? Sad as well, because sometimes we would see the chicks playing with the paint chips and like in their beak, and I'd run over there, and you're like trying to get the chips out of the chick's beak because you knew that they really shouldn't eat them. So it was something that we actually witnessed as well, them playing around with those paint chips. So we published the work, and then I thought, okay, that's it. They're going to clean up this problem, right? So there's paint on the buildings, and it's sort of an easy fix. It's kind of expensive because it's Midway, it's far away, but we should fix this. But then it kind of came down to, well, who's going to pay for it? And really, should we do this? And is lead poisoning really a priority for action? And when I talked to different nonprofit organizations that were involved with albatross conservation, they would tell me that, well, we're spending all of our conservation dollars on trying to reduce fisheries bycatch. So albatross, like many other marine species, will be killed accidentally as bycatch in different fishing operations. This is actually a picture of a wandering albatross being submerged on a line, but it's a nice illustrative photo, so it's one that's used often. And fisheries bycatch is a primary threat to albatross survival across the globe. And so nonprofit organizations didn't have extra conservation dollars to deal with the lead-based paint problem. And trying to find then resources or just like understanding whether or not it was a priority to be fixed was hindering the ability to do something about this problem. So it became clear that we needed to quantify what the population level impact of lead was to these to Laysan albatross to prioritize conservation action. So you can do this using something called a population viability analysis, which is basically just a mathematical model to predict the likely future status of a population. And they're used a lot in conservation literature and for understanding, like prioritizing different conservation actions. They can be used to help tell you how many grizzly bears do you need to make sure you have some in the future. They can be used to tell you that you need to put like these turtle excluder devices on your shrimp nets so you don't catch loggerhead sea turtles, or how much habitat do you need to save spotted owls. So they're a very common tool, and they help measure like an impact to populations. So I did a population viability analysis to understand the impact of lead on Laysan albatross. So what goes into the population viability analysis is survivorship of individuals by age and the number of young per female, which for albatross is just 1. And then we've also put in impacts. So I counted all the buildings on the island and quantified the number of chicks that are most likely, exposed to lead, and determined that about 10,000 chicks per year were likely lethally exposed to lead-based paint on Midway. And we also quantified impacts from bycatch on adult survival. So we could compare sort of the difference between lead poisoning and bycatch on population health in the same model. And so when I did this work, the projected Midway albatross population was declining. And on the y-axis is population size, and the x is years into the future. So you get these projections out. So current conditions was a declining population. And then we could say, well, what if you eliminate lead deaths from the population and you get a bump up? It's still declining, but not quite as severe. But then this lets you show that you also really need to increase adult survivorship by 1% to get the population back up to stable. So you need a combination of reducing bycatch and reducing lead deaths together to help, buffer the population. But these models really help you do this, and you can then basically understand the impact and put it kind of together. And for this, we could say, well, okay, we know fisheries bycatch is a problem, and we really need to work on reducing that. But in the meantime, we could eliminate lead poisoning today and have close to 200,000 more birds in 50 years, help buffer against these impacts from fisheries bycatch, which is a lot more, it's just a lot more difficult of a problem to deal with. So publish the work again. And also realize no one really reads your published paper. So what you need to do is also talk to people that are involved. We talked to actually the Department of Defense because it was, the military's lead-based paint on those islands. We worked with different nonprofit groups. I worked a lot with the Fish and Wildlife Service, met with the Department of Interior, trying to understand this. Still was having a hard time getting traction to do something about this issue. Audubon, California became involved, sent out a call to their members to try to do something about this. The Center for Biological Diversity actually threatened to sue. It's a really sad, tragic thing to see these lead poisoned chicks that are just destined to die. I actually watched one dying chick one day, like when I was out there, and a parent spent hours trying to coax its chick to feed, and it's heart-wrenching. So I worked on this issue for probably like 10 years, but eventually the Fish and Wildlife Service, they submitted a petition to list this as a Superfund site, which opened up a different type of funding. And so in June 2010, the lead-based paint cleanup was authorized by the Department of Interior, and it was completed October 2017. So it's huge for me, a huge win, and saving probably at least 10,000 Laysan albatross chicks a year and other birds that could be out there. We were worried about the bone-in petrels burrowing there. So it's a great solution, and it really feels good that this got done. And now condors. So that was my Laysan albatross study. So now I'm going to talk to you about condors. They're North America's largest terrestrial bird. They can also live a long time. They have a very interesting and similar life history strategy to Laysan albatross, and that's just coincidental, I swear. They have low reproduction as well, but they have about one chick every other year. So I love this picture. It was taken by somebody in the Tehachapi Mountains, I think. And condors are really social and curious. And I heard that after, right after this picture was taken, this dog ran out and got its ducky to then show the condor who's just hanging out looking at it. But condors are really interesting and cool creatures, very curious and social. And they're also one of the world's rarest birds. In the 1980s, there was only 22 condors, all in captivity. In 1992 was the first release of captive reared birds back to the wild. So the release of captive bred condors has been responsible for the wild population increase. And in fact, the California Condor recovery of the wild population is touted as a huge victory for endangered species management, of which it should be. It's a really phenomenal feat. On the y-axis here is the number of birds, and then years, and this is just the total number of wild birds, and you can see them increasing. But this actual story is a little bit more complicated. So here, these lines are cumulative numbers. So the blue here is total release, total dead or removed, and the blue dotted is total wild fledges. And you can see here that more die or removed than are born. And so this population is not sustainable in the wild. And why is that the case? So between the 1970s and 2000s, there was limited evidence that lead is a problem for California condors. And then from the 1990s to today, they are monitored on a near daily basis, and their blood lead is checked about twice per year. Pretty much all condors are tagged with and fitted with radio. A lot of them also have GPS or GSM tags. And so they're checked every day. They're brought into, they're handled twice a year for their blood lead to be taken. And every day when they're monitored, if they seem to be acting weird or something's happening, they'll go in there and try to trap them and do a health check to assess them. So they're very closely monitored. And unlike albatross, I mean, the entire population is known. It's all got a stud book, they're all numbered, and everything like that. And because of that, we have a lot of data on these birds. So on the y-axis now is the percent lead poisoned. And here I'm calling lead poisoned a blood lead above about 45 microgram per deciliter, which would trigger chelation or medical intervention for California condors. As you can see here, the percent that's lead poisoned from the 1990s to about 2013. And the bottom line is about 20% of condors that are tested per year need clinical treatment, which is a huge number, right? These birds are poisoned to epidemic proportions. But to me, which is even kind of more alarming, is about 60% of wild birds out there have been lead poisoned at least once. And many condors have been lead poisoned many times in their lifetime, sometimes many times during a year. So they're lead poisoned constantly. And then some of the work that we've done with feathers show that it's actually much worse than the blood lead indicates. Blood really only tells you about 2 weeks of exposure, and a feather can show you about 4 months of exposure. And I'll illustrate how you do this. So we've determined that about one piece of feather is about 2 centimeters, represents to about 4 to 5 days of growth. So on the y-axis is estimated blood lead, and then on the x-axis is the timeline over feather growth. So this is basically showing you the whole feather lead profile of 2 feathers actually from a condor that was released in January and then died in July that same year. So you can see here these peaks, these 4 peaks represent 4 separate lead poisoning events that the condor had during its brief lifetime in the wild. Now this condor, like all condors, was monitored regularly, and they actually did catch one of these lead poisoning events at the beginning. The condor was brought in for medical treatment. It was chelated to get the lead out of the body, and then released back into the wild where it went and got lead poisoned again. Blood lead went down pretty quickly, lead poisoned again, and then one more time and the condor died. So these feathers provide a lot of information about the history of lead exposure throughout this 4 months of feather growth. And so if you just kind of look at it another way, so blood again reflects about 2 weeks of exposure. On the y-axis now is percent condors. And this is proportion that are exposed, poisoned, or lethal. Lethal here is over 100 microgram per deciliter. Exposed is over 10. And you can see that when you look at the feathers, it shows that the lead poisoning is about 5 times higher than what we thought from the blood. And so we see this time and time again, that lead poisoning is much worse, much more frequent, happens much more often to much higher levels than the blood data indicate. So it's not surprising then to know that lead poisoning is actually the #1 mortality factor for juvenile and adult free-flying condors. And so this is a really big problem, right? So we know that condors ingest spent lead ammunition. This is a radiograph of a rifle-killed coyote, and you can see all these basically radio-positive images here are all little pieces of lead like shrapnel throughout the carcass. Condors then will feed on the carcass, and hopefully you can see this tiny little radiopositive piece right here is a little lead fragment that poisoned this bird. So just one of these small shrapnel pieces, if the condor ingests it, it can poison and potentially kill the bird. So it doesn't take much lead at all. And you can see why it's such a big problem then. And so then this lead poisoned condor becomes sick and then brought in for treatment. And so some cases we actually have the lead item because the condor is brought in for treatment and they are able to recover the lead item. So I can look at the isotopic signature similar to what we did with Laysan and Albatross to show that the lead item is a source of lead to the condors. So here's another radiograph of a condor. This is 401, and actually this bird ingested a lead buckshot, which is a little easier to see here, this buckshot. And so this is the lead ratio on the y-axis. This is the lead item in the blood. And again, you can see that they're not measurably different. So we looked across different condors that had ingested lead items, and in every case, the lead item matched the isotopic signature of the lead item, was not measurably different than the isotopic signature of the blood, showing that that lead item poisoned the bird. So this was helpful and shows that a condor that will eat a lead buckshot is poisoned by a lead buckshot, which is not surprising, but was good to show. But it's also limited because in many cases, they don't recover the lead item. So we also did a larger study where we basically looked at condor blood and ammunition samples in a large kind of free-flying study. We looked at pre-release condors for background lead, so blood lead from birds before they were released into the wild, blood lead of wild condors, lead-based ammunition that we got from hunter exchange programs and also purchased at stores throughout California, and quantitative modeling. So on the y-axis, again, is this lead ratio signature, and on the x-axis now is blood lead, microgram per deciliter, in log scale. So first to show you, this is the pre-release background signature. So we can measure very low levels of lead in organisms and get an isotopic signature. So even though these levels are low, they're all under 10, we still can get what is the isotopic signature. Now for this pre-release background, this actually matches sort of background lead in California, what we used to have in our gasoline tanks actually. And it matches, like if you were gonna measure lichen on gravestones or something. This is sort of just background California lead. So we also did 76 ammunition samples, and it had a large range. So again, this was from different sources through hunter exchange programs and also through purchasing at retail stores in California. But first to show you that overall, the background and the ammo did have different isotopic signatures. And then you can overlay that with the wild birds, and you can see that they have an isotopic signature that basically falls between sort of pre-release and ammo, with in general, as the blood lead getting higher, the bird's more and more likely to look like ammunition and less likely to look like pre-release background. So as it gets more and more, they're looking more like ammunition. And so with quantitative modeling with these data, we could show that lead in wild birds, about 80% of the lead in wild birds is best explained by ammunition. And you might want to know what these 5 little dots up here is. Actually, this is paint. So condors do sometimes eat paint. In a very limited case, this was paint on an old fire watch tower at Pinnacles National Park. But as soon as this happened, a couple of birds were poisoned. We were able to identify the source right away, and immediately they went and abated this lead paint source and dealt with this problem. So this was a very, what's the word? It's a very a small kind of like constrained source, but it was able to be fixed quickly. But it just shows you that this isotopic analysis can work. It's not only just ammunition, you can use it for other things as well. So we also did population viability analysis to understand what the lead poisoning impact is on California condors. And all condors are tracked, so this was pretty easy. We could look at the annual survivorship of individuals and the number of fledged per young per female, so the actual survivorship and reproduction from the birds that are out there. And then for these models, we assumed no future releases, and we looked at 3 scenarios. Continued management for lead, which is what they do. If they treat them if they're sick, they also do other supportive care. So all the kind of management that's being done now continues. We could look at cessation of management for lead poisoning. So what happens when a condor hits a lethal lead level and dies and isn't treated? What would happen to the population? And we could also look at what happens if you get rid of lead deaths for the population. How will the condors do? So on the y-axis is the free-flying birds in California and then years into the future. And with this model, if you look at lead condors without any future releases, the population is basically just the same with lead management. You're not seeing an increase. So reinforcing that the increased growth of the condor population is driven by those captive reared releases going into the wild population. And then if you get rid of lead management and then the condors start to die at these high lead levels, you can see that quickly then the condor population plummets again close towards extinction. And so this was really important to show that, lead is still a problem out there. And if these condors still, start dying at higher rates from lead, then the population will, go down close to extinction again. But if you get rid of lead, or if you greatly reduce lead deaths, it shows that the condor population can be self-sustaining and can increase. And that was very encouraging for the condor managers to see. So the big thing from this model is that lead is impacting condor recovery, but if lead-related mortality is reduced or greatly reduced or eliminated, condors could recover. But one thing to know is that one carcass with lead can kill many condors. So this is a camera footage of a feeding station, and this is a cow that's staked out there, and the timestamp is 2:02. Just want to show you that. And so at 2:58, the condors come in and they're like, supper's on, and they come in to start feeding. 3:10, a bunch of condors come in. And when this photo was taken, the condors in here represent about half the free-flying population of California. So you can think one carcass really could poison a lot of condors. And then at 4:40, the only thing left is the bone that's staked to the ground. So it shows you pretty quickly how this, lead poisoning of just a few carcasses out there can be a problem. And we wanted to illustrate that quantitatively. So we looked at this and basically how big of a problem is just a few carcasses on the landscape. So on the y-axis here is the annual probability of lead exposure, and on the x-axis is the percent carcasses with lead. And we determined that condors probably encounter, depending on if the carcasses are small or large, between 75 and 150 carcasses per year. So if only 1% of carcasses on the landscape have lead, a condor still has a 30 to 50% chance of annual exposure per year, if only 1% has lead. But condors have to live a really long time, right? So over 10 years, if only 0.5% of carcasses on the landscape have lead, a condor still has close to 100% chance of exposure, 85 to 98% chance. And I think this illustrates just really how just a few contaminated carcasses on the landscape can be a really big problem for condors. So we published this work in the journal of the Proceedings of the National Academy of Sciences, and which was exciting. And they put a picture of a condor on the cover, which was exciting. But again, nobody reads very few people read your scientific papers. And we thought that this was such a big issue, and the health risks from lead-based ammunition is such a big issue, not only for condors, but for other wildlife and for humans as well, that we got together and we put together a consensus statement of scientists with 30 experts on health effects of lead and talking about the health risks for lead-based ammunition. We published our perspectives in a journal, plus put out this consensus statement, and then did a bunch of outreach to our partners and other stakeholders that were involved. My collaborator, Don Smith, and I both testified before the California Senate on this issue. And we were very excited to see that in 2013, Governor Jerry Brown signed the law of AB 711, which is basically requiring California law to require non-lead ammunition for hunting. It had a long phase-in, but the phase-in was completed July 1st, 2019. But California is actually the only state in the nation to outlaw or to require non-lead ammunition for hunting. We were thinking that other states might also pass similar legislation after 2013, but no other state did. So California is still the only state in the nation that requires this. Which is kind of a problem because lead is still killing. So these are screen grabs from news sites just this year. Lead poisoning is killing an unusually high number of California condors. They're still being lead poisoned. This is from a different state. Eagle poisoned to death, a hunter's call for unleaded ammo. So lead is still a problem. And then I'll just sort of revert you back to the quote I said at the beginning. We've known this has been a problem for over 200 years, and we have a solution to fix it. I just feel like we need to implement those solutions. So I want to help generate information to do that. And so for the end of the talk here, I'm just going to talk to you a little bit about some of my current projects. So California condors are not out of the woods yet, unfortunately. And we are working a lot with the Fish and Wildlife Service, U.S. Fish and Wildlife Service, and the Condor Recovery Team in general to help generate models to aid in condor conservation. Specifically, we're doing population viability analyses to understand how different management actions will affect condor survival. And I'm going to just give you one example today of what we're doing. So we're working with a much more complicated model than what I showed you that we published in the paper I just showed you. This model has successful breeders, failed breeders, widows, skippers, all kinds of things. All these lines are probability of transitioning from one stage to another. The one thing that's really important with this model is it includes these captive bred releases into the model. So these are the captive bred releases. They have a different survivorship at first, and then they enter into the main model. And the reason that this is so important is because this is the reality for condors, right? They are captive bred, they're releasing captive bred birds every year. And so we had to be able to incorporate that in the models. So some of the work that we're doing is we're doing these models to help inform habitat conservation plans for wind energy facilities. So you have condors, and theoretically they could hit a wind energy facility and you could lose some condors. And so for these habitat conservation plans, they're trying to figure out how do we mitigate or offset the potential for a condor to be killed. I'd like it to be known that so far no condor has ever been killed in a wind energy facility, but the possibility exists. And so the Fish and Wildlife Service is trying to prepare for that. So can't we just replace it? Now, can't we just put 2 captive bred juveniles into the population if 2 adult condors were killed? But it doesn't mathematically work that way. A captive bred 1.5-year-old doesn't contribute to population growth as much as a lost breeder, because it takes at least 5 years for that 1.5-year-old to then, they don't become a breeder yet. So we can look at this using the model. So to create something that we call a replacement ratio, and this is how much would you need to replace if you lost a breeder. So on the y-axis is the change in number of females, and this is years. So basically you can model with, on the black here is no wind mortality, and then you model that birds get taken out of the population, and then you put birds back in, and you want to basically get it back up to basically no mortality. You want to get it back to baseline. So this is no replacement, so that's too far down. And you can see here's 1-to-1, so that's also too far down. It doesn't get you back up to where you need to be. But we determined that basically you had to have, the loss of an adult the loss of an adult condor could be offset by the release of 2 to 3 captive bred 1.5-year-old juveniles. So this is some way that we can use these models to basically quantify what this replacement ratio is so that you can offset any potential mortality from wind. So that's just one example of some of the population model work that we're doing with the California Condor recovery team now. And then I just wanted to finish with something I'm doing with golden eagles. So I'm really passionate about lead, as you can probably tell. And I've been working on this issue for a long time, from the albatross to now the condors. And to me, it seems like there's a lack of information on the population level impact of lead poisoning to other species, which is a barrier to finding these solutions. So we know golden eagles occur throughout the continental United States, and we also know golden eagles are lead poisoned. So I've been doing work on golden eagle feather lead to try to understand what the lead exposure rates are in golden eagles, and have found that similar to condors, feathers show that a high proportion of eagles are lead exposed. And we found that about 40% of eagles that we have looked at have been lead poisoned from looking at the feathers. Another thing we can do, similar to the condors, is we can use these feathers to basically reconstruct the lead exposure history over the time frame of the eagle's feather that grew. So I'll show you one example of that. So on the y-axis, here's feather lead in microgram per gram now. And so this is a feather lead profile from one feather of one eagle that was found dead at a wind energy facility. Wind energy facility, it's getting late, huh? And the feather lead profile illustrates that this bird was lead exposed at the time of death. So the y-axis is estimated days before death. So this is 34 days before death, about 20 days, now the feather lead is going up. The bird's been exposed. Between here, it's going up, and then the bird dies. And it died from a collision at a wind energy facility. And one of the things that we think about, because wind energy collisions, for golden eagles is a problem, and wondering if there's a relationship between lead and collisions. So are birds that are lead exposed more likely to hit a wind turbine? We don't know what the answer is, but it's something that I'm really interested in looking at. So for golden eagle lead poisoning, the goal is to quantify, or what our objective is to quantify lead poisoning rates using feathers, because we think they're a really powerful tool. And with condors, we've really come to learn that the blood monitoring data is just not very sensitive. It's not very good at understanding lead exposure rates. We also want to quantify the impact to the population and to provide data to help inform a solution for lead poisoning in other avian scavenging species. And with that, I just wanted to thank, this research has spanned like 20 years that I've talked about. So lots and lots of people have helped, and lots of agencies and foundations have funded this work. Lots of photo credits in there. So I wanted to acknowledge that. And I just wanted to end with, this is a great website, and it's a really great resource. The people that run this website are very, very informative. So if you have any questions about hunting with non-lead, or if you have any friends or family that have questions, I really encourage you to go to this site and to check it out. And if you want to contact them or contact me, and I'll put you in touch with them. They're awesome, they're great, they know a lot. And with that, I'd like to thank you for your time.
Myra, that was absolutely wonderful. Thank you very, very much.
Yeah.
Thank you so much. That was fantastic.
And
You have lots of questions too,
it has generated lots of questions.
which is great. So our first question we have from James Zimmer on Midway. Has there been a follow-up study demonstrating the absence of droop wing, I guess, since the lead paint has been removed?
That's a great question. I tried to get someone to pay for me to go back to do that. But no, I did have some friends that were out there, and they did look around, and it looked like drooping was much less. So, but it was not done in any quantitative, systematic way. It was just, hey, you're going out there, can you look for me? So it did look like it. I mean, the thing about it being cleaned by the Department of the Interior, like being designated, one of those sites, is they had to clean it to a special level. So I felt pretty good about the cleanup. I talked to some of the consultants that were working on it, and I felt really good about it. So I've heard anecdotally that it's less, and I'd love the chance to go out there and do a follow-up study, but yeah, haven't had the chance.
Okay, great, thank you. Lance asks, which other bird species are severely affected by lead poisoning? He asks about common loons. And what about other species on Midway other than the Laysan albatross?
Yeah, so for Midway, we worried a bit about the petrels because they would burrow under the buildings. That black-footed nest there as well, but they didn't nest right next to the buildings. The Laysan albatross were right next to the building, so it definitely seemed to be something that was affecting them the worst. We worried about people too, because people were out there sort of like scraping paint off. So for Midway, Laysan albatross were the pretty much the main species that we found that seemed to be sort of interacting with the paint. For lead, like any scavenging species can be affected by lead from lead-based ammunition. Bald eagles are also impacted, turkey vultures, ravens, red-tailed hawks. I mean, anything that scavenges can do it. You can think of, mountain lions, coyotes, anything. And humans that eat game shot with lead-based ammunition can also get elevated lead levels. So it's a problem for humans as well. Hopefully I answered that kind of going around a bit.
Yeah, yes. Thank you. Let's see. Mike asks, do other California populations like in Baja or Utah suffer from similar lead exposure and mortality than the ones in California?
So yes, for the Utah, Arizona, we're actually just working on a global condor population model right now where we're incorporating data from all of the different populations. So from the Arizona, Utah, and the Baja and California populations. But unfortunately, yes, they still have lead poisoning is a really big problem for the condors that are in the Arizona, Utah. It seems to be less for Baja, so we're still looking at that. But Baja doesn't have the same level. My understanding is they don't, have the same level of hunting that we do in California, and the condors in Baja tend to have lower lead poisoning rates. So we kind of wonder whether or not Baja's just like going to be a great place for them because they're doing pretty good. It's pretty hard to track them. The terrain's really rough, and they're not as closely monitored as the California and Utah, Arizona population. But I would say, to answer your question in a roundabout way, yes, for the continental US, those condors are lead poisoned. But Baja looks like those birds might have lower lead poisoning, lower lead exposure rate.
Okay, great. James Zimmer asks again, let's see, the midway birds with lead, that have lead and they do survive, do they rinse out the lead or can lead from the blood of the adult be passed to the egg when the egg is laid?
Yeah, so the adults were all really low. So they didn't seem to have, we didn't see any adults that had any elevated lead. Unfortunately, that the lead levels, especially once they would get peripheral neuropathy, they were moribund. They were not going to survive. They can't fly. If they did have a lower level, but when you ingest a piece of paint, there's not really we didn't see that many. We kind of saw they were either really lead poisoned and they had droop wing, or they were away from the buildings and they weren't exposed. We know that lead has lots of other sublethal effects. So for our modeling, we assumed if they were kind of lead poisoned around the buildings, they weren't going to make it to come back into the population. But we talked, like people asked me, well, could you chelate them? Could you help them? But once you have peripheral, like the damaged nerves like that, they're not, they weren't going to survive.
Okay, yeah. Thomas Walton asks, are condors also poisoned by any other types of ammunition?
So that's a great question. Not that we've seen. So they have been, there has been cases of I want to say now I know this is being recorded. So zinc or something from, they think from coins. So they think there's been something of them picking up coins or micro trash or something. We haven't seen evidence that they've been poisoned by other types of ammunition. So there's no evidence that they're poisoned by copper ammunition or anything else. So we haven't seen it. Just lead.
Okay, thank you. Do condors go to the ground under a windmill to eat birds that have been killed?
I don't know.
That's a fair question.
I've never seen that. I don't know. I could ask. I've never, I don't know. I don't think I've ever seen that. They have a lot of the, because a lot of the birds have tags as well. So the wind mills will, they'll shut the wind turbines down if a condor becomes near it. A lot of them have these geofences.
Oh, really? I didn't know.
Yeah.
Okay, the wind farms know where the condors are.
Some of them do, but then I worry, what about the eagles and the bats, right? They don't have the same.
Right.
And no, I've never heard of them feeding on animals. I'll ask.
Yeah. Do condors actually eat birds? I mean, there's not, or usually larger things like dead deer and.
Yeah, my understanding is there hasn't been a documented case of them eating another avian species, but maybe they would, but I haven't seen it.
Okay.
Then you say they're obligate scavengers completely, but then there's this one documented case of them feeding on a not quite dead sea lion pup once. So there's always trying to, no absolutes in nature.
Mm-hmm.
Myra, what about condors feeding on marine mammals? You know, because there's clearly a lot of those have high levels of PCBs and DDEs and other sorts of persistent organics in their
Yeah.
fat. So does that, have you measured that in condors, and does it make lead poisoning worse?
Well, we have measured that. I could have done the condor contaminant talk and talked about that, but I didn't do that. So condors are getting exposed to high levels of DDE, which is the main breakdown product for the agricultural pesticide DDT from feeding on marine mammals, California sea lions specifically. So that is a problem, and we think it's lowering their reproductive success. Ventana Wildlife Society has shown that they have thinner eggs on the condors that, like the coastal condors in Central California. So that is a problem for reproduction. We haven't seen that there's a synergistic impact between lead and DDE. In fact, condors that tend to feed on the coast and feed on marine mammals tend to have lower lead poisoning than the ones that don't. So we think they're kind of, you know, those marine mammals tend to not be shot with lead. So it's kind of like been that way. But it is kind of a problem for condors as well. It's not as big of a problem as lead, but it is something that is impacting a little bit the reproductive success for the ones that are feeding along the coast here in Central California.
Okay, Lily, one of our VabS members asked, has there been droopwing in California condors?
So we have not, so they do get different kinds of paralysis. So they will get, so they'll get like where they can't stand up and other types of things. The actual droopwing, I don't know if it's sort of a developmental thing that the chicks get, because it's a very kind of classic, you know, different thing. So I would say not the classic kind of manifestation that we saw in the Laysan albatross chicks, but condors do get paralysis of different types of things from lead poisoning.
And yeah, and I get, but sometimes I guess it's not permanent, like in the droopwings. You can treat them, and I guess, is that just catching it early, or?
Yeah, I mean, you can treat them, but a lot of times once they start with that far gone, it is hard to bring them back.
Okay.
I mean, sometimes I think just because they're so phenomenal at the LA Zoo, right? Like they're just amazing that they can bring these birds back. But it's not I think once they start having different types of paralysis and things, it's pretty hard.
Barbara asked, what could be considered a very obvious question that we'd all want to ask, and very politically charged. So why can't we just replace all hunting ammunition with non-lead?
I don't know. I wish we could. I mean, I think it's to me, it just makes sense from a human health and wildlife health perspective, because these alternatives exist and they work. But I think it has become an issue that people don't want to be told what they can use to shoot with, and they don't. There's been a lot of question about the science. You know, when no one questioned that lead paint was a source of poisoning when we did that work. I mean, everyone agreed with it, like the studies. And some people said, we just don't have the money, we can't fix it. And maybe they questioned how important is it for the population, because it's a chick, not an adult. But they didn't question the science. But when the lead worked, there was a lot more, still is actually, questioning about it's not, lead can't do that. There's different forms of lead, like just weird things that I hear. That's not the case. And I don't really understand why. It just seems that it became something that people decided to be polarized about.
Is it more expensive to have, say, copper ammunition? Is that part of the problem, or?
So that's something that people say. There's a paper that got published that says if you use the same quality ammunition, then it's the same price. But I think there are cheaper lead options out there. Maybe there has been a case of like, there's been a problem with like availability of different types. Like there's certain types of non-lead shot that was hard to find in different types of ammo, but that's becoming better. And I think the price point difference is really, I mean, I've heard it's really small. So it's not like that's not the barrier, even though people sometimes talk about it. I know there's a lot of non-lead ammo giveaways that people are doing to try to help facilitate it and to educate them. And again, because you would never want to feed your child meat that was shot with lead ammunition if they have no safe level of exposure for a kid. I just anyway.
Yeah.
Interesting question by John LaBelle. Can you speak about the correlation between the ban of leaded gasoline and the drop in the crime rate 23 years later?
Interesting, huh? Well, no, but there are studies that have shown that lead exposure. There is a correlation between lead exposure and aggressive behavior or things like that. So lead does all kinds of things. So I just.
What about this is a question from Lance? What about the use of lead fishing tackle in California? Is it still in use? Some states are banning it to protect species.
Yeah, that was not covered under AB 711. It was discussed, I think, but it didn't seem like something that was able to be covered. So it is still in use. It's also a problem. Yeah, anytime that you're putting large amounts of lead into the environment where it can be ingested by wild animals, wildlife is a bad thing.
Mm-hmm.
And
maybe in this case for something like bald eagles that might, eat some, although, it's not the same as shot where they're embedded in the food. Okay. Great. Rich asks, there are 2 records of condors passing through the Altamont Pass wind resource area in eastern Alameda County. There have been concern expressed in public meetings and Zoom presentations, presumably about them being hit.
Interesting.
More of a comment. Morgan asks, how is the lead removed from the condor's body when they treat them?
So they give them these drugs. That's how they give people as well, these chelation drugs that bind to lead in the blood. So it basically binds up lead and will help remove it from your blood. And then you usually have to go through multiple courses. And then you have lead that's also like maybe in your soft tissues, like your liver and etc. So once you get it out of your blood, you'll get a rebound. It comes back up again. So you have to, it's not this benign treatment, because also the drugs that bind to lead bind to calcium and other essential things that you need. So you have to give those back. So there's like 2 different main types of drugs that you use. You can use EDTA and you can also use Suxamer. But for condors, they use calcium EDTA as a way to basically try to pull the lead out of the bird's body. But it's not a benign thing.
It's not, not just, hey, give them a pill and everything's fine.
You have to monitor it.
Robin asks if you checked lead levels in king and Andean condors in South America.
I have not. I've talked to people that have worked with Andean condors, and I think that it is a problem, or they're worried about it, but I'm not sure. I guess I'm not sure how much data they have on like understanding like the frequency of lead poisoning, but I know they're worried about Andean condors for, you know, different kinds of things. But I have not.
Rich has another comment. Switching from lead to brass ammo requires a new barrel. So maybe that's a reason that at least for brass. I'm not sure if that's the same as copper or any of the other ones, but
I've been told that you have to recite and stuff, so it's not as easy as just like I'm not a hunter, so I don't know, but I've been told it's not as easy as just like switching a different bullet in. You have to do this stuff, so that's another barrier. Like you have to, you know, recite and do these things. So there are.
So it's a little bit of a pain. It's not just, you know, hey, buy these bullets instead of these, and everything's the same.
You gotta, yeah. Okay. You gotta kind of make the switch, I think.
Barbara asks, I thought people hunt to eat the game. Why would you eat something hunted with lead?
Yeah.
Yeah.
I'm not
sure
know.
if there's an answer to that. Thomas asked, can a condor die from the chelation process?
So I should say it has happened that there's been complications where they've died and unsure what type, what was it specifically? Was it a combination of the chelation treatment, the lead poisoning, and something else? You know, they're in the zoo setting. So some condors sometimes don't make it through. There's been a couple of instances where they felt that might have triggered some kind of a renal failure or something, but it's hard to tease out. They're already lead poisoned.
Hard to figure out exactly what.
Yeah, we've had an awful lot of chat. Thanking you for a wonderful, wonderful presentation. But we did have one question. I know with condors, it's much easier to get data and things like that. Have you heard about lead poisoning in turkey vultures?
Yeah, so a little bit. They there's been a few studies looking at lead poisoning in turkey vultures, and I definitely think it happens. And studies have shown that it happens. And so whether or not it's a problem at the population level is not, probably not, because they're doing much better.
Yeah.
But basically any scavenging species, I think, is most likely, some individuals have probably died from lead poisoning.
Yeah, which makes total sense, of course. Well, Myra, your talk has generated tons and tons of chat, and it's all wonderful. And I can summarize it up by saying, yeah, it was a wonderful, wonderful presentation. We all enjoyed it. Thank you very, very much.
And one last
thank
thing.
you for all the work that you're doing and how
Yes.
much it helps saving condors and Laysan albatrosses and anything else, golden eagles in the future, I'm sure.
And you can make donations to support this kind of work through Myra's lab. Can you tell us how to do that, Myra?
There's the university there. We have a foundation, so there's a link that if people want to, they can donate to my research program using this link that I could put up, I guess, or something.
I think we have it here.
Is this the right link here on the slide?
Yeah. It should be it, I think. Is it?
Yeah, it is.
It could be a bit longer than that because you just need to go to my specific research program.
Oh. Yeah, that's just probably it lists all the different ways you can donate, all the different programs at UCSC.
We can put it on the website, Myra, the correct long one.
So please send it.
Just send us what the link is, and
yeah, we'll put it up under the recording of this webinar.
Yeah.
Buy some pipette tips, then, or I'll be swimming out to the barge.
One of course, very important question is, what kind of cat do you have?
Oh, you can hear him?
I thought it was our cat.
That's enough. vocal animal. He doesn't. They don't like being locked out of the room.
No, they don't. They're very social animals. Well, once again, Myra, thank you very, very much. It was a wonderful webinar. Everyone really enjoyed it. And
Wow.
I hope everyone can join us next week for parrots. It's that also is something that actually we all deal with, and we'll have a field trip the following Saturday. And we have lots of wonderful things coming up. Please check out our website at labirders.org for other things coming up. And with that, thank you again, Myra. And I will,
Thank you, Myra.
Thank you, Myra.
Thank you, Myra.
Thank you.
and we'll see you all next week. Take
Good
care,
night,
everyone.
everybody.
Bye.
Good night. We'll be in
Bye.
touch, Myra.
Bye.
Thank you.
Bye.
Bye-bye.